actually starts with glycolysis. It's the portion of a small yield, it's the production of a small yield of ATP without the use of any oxygen by converting sugar into pyruvate. So this actually happens in the cytoplasm of a cell, not the mitochondria. So we're still in the cytoplasm of the cell itself. So ATP is used by phosphorylating sugar. So the ATP here, it kind of has to be made somehow. So we have fructose biphosphate. That's made when ADP is converted into ATP.
Yeah, I'm filming Caleb first.
So when these two split, it forms fructose biphosphate. And now you have triose biphosphate. Once that splits. These three trio biphosphate are now oxidized to create glycerin 3-phosphate. This reaction of the oxidation yields enough energy to make ATP. Okay. So we also need to make NADH and able to move our electrons later on. Now remember that hydrogens carry 2 electrons, 2 negative electrons per ion. So, to make NADH, it's made in a chain reaction into the splitting of the ATP and the ADP that we've already done. So, when this ADP reaction happens, hydrogen ions are removed. Now, if those ions stay removed, then there's no electrons and no oxidation. Instead, the hydrogens are accepted by NAD. And when they are, it's turned into NADH. So, now in these last stages, the last phosphate group that's still here is transferred to ADP to create ATP. And pyruvate. This happens twice for glucose.
Yeah, that sounds right.
Okay, so to show that this is like a glycolysis reaction, one glucose molecule enters a cell's cytoplasm, and after 11 steps that take too long to explain, the 6-carbon molecule splits into two 3-carbon molecules that we have named pyruvate. Now, when that happens, if oxygen is present, it's absorbed into the mitochondria where it's fully oxidized. Carbon and oxygen are then removed in the forms of CO2 as a product of glycolysis. This is called decarboxylation. So pyruvate oxidation is also done with the removal of hydrogen atom pairs. Then the hydrogen carriers, NAD and FAD, accept the hydrogen atoms and pass them on to the electron transport chain, which we will talk about later. So now we're get on to the link reaction. So when pyruvate diffuses into the mitochondria, it must be decarboxylated, which I explained earlier. So this is done with an enzyme named coenzyme A. So once the pyruvate is completely decarboxylated by coenzyme A, it creates acetyl-CoA, which is necessary for the Krebs cycle.